Hi, in this video we are going to be discussing mutations and genetic disorders. Okay, so first let us remind you of the setup. And the setup is that you have the cells, and the cells have chromosomes inside them, again 23 pairs. And then the chromosome is really just a packaging for the DNA. So what happens is the DNA is so long that you need to wrap it around histones, and the histones form nucleosomes, and then those nucleosomes form the what would become the chromosomes, like so 23 of them. And then next is I want to show you what mutations happen during the replication and during replication or protein synthesis. So what happens is when you base pair, right? Sometimes a letter is inserted, right? So this is now on the level of a nucleotide basis. Sometimes a letter is inserted. So instead of it is just GAGAC TTAC, what happens is a letter could be inserted, right? So you have now changed the code. Sometimes it could be a substitution, meaning it should be C here, but it's substituted with an A, such that the code changes. And then you could have a deletion where there should be a C there, but it's deleted. Okay, so let's take a look at why that happens. Well, again, you have the DNA is split into two, right? If we're talking about the replication part, let's just take a look at the leading strands because it's easier. So, for example, if this is a C A T like that, so it base pair. So this one is base pair with C because that's a G, and the C would be paired with the G, and the A would be paired with a T, and the T would be paired with an A. But sometimes, again, the machinery makes mistakes. So instead of a T there, you put a G, for example, and then the rest has no mistake. There's only one mistake here, and then you say, I thought, sir, you discussed that G and T will not bond because they don't have the number of bonds and they don't have the right pairing. Yes, but again, you could force the issue. But what happens is when you force that issue, well, I mean, when you force uh, G to bond with T, again, this area of the DNA will not be smooth because there will be some misalignment, for example, and so on and so forth. So what happens is this continues, and then there is a checking mechanism. There's a checking mechanism in the DNA. So, for example, the ligase, before it seals up this DNA, it will check if the pairings are correct, right? So, what happens is most of the time it catches this error and says, "Oh, oops, this is a T. This should be an A, not a G." So we replace this and replace it with an A, and there's no mutation that happens. But the thing here is the DNA actually doesn't know. Once the two strands are created, the DNA doesn't know, or at least its mechanism is not aware of which one of that is a mistake. So what happens is, okay, they see that this is a T paired with a G. They know that this should not be the thing that should be paired, but the DNA doesn't know if which one is correct there. Is it the T that's correct and G is wrong? Or is it G that is correct and T is wrong? Okay? If the DNA makes the correct decision and says, okay, well, let's replace this with an A, then there's no mutation because that's the original setup anyways. But if the DNA makes a mistake and assumes that this is the correct one and replaces this instead, then you have a mutation because remember this is supposed to be T A, but now because again the DNA thought that this is the correct one and this is the wrong one, so it keeps the G here and replaces with C. So in this case, you have a mutation, and that mutation is a substitution, right? Because this pairing here should be AT, or sorry, TA, should be TA, but now gets replaced. It's now CG. right? So those things could happen. It could also happen that when the when the checking happens, the DNA would say, this is wrong, so let's just cut it out. So if you cut that out, then one letter is deleted, so that's a deletion, right? That's a deletion. The first one that we discussed is substitution, and then you could have a deletion, and then you could have an insertion, meaning you have T, A, C, G, for example, and then this base pairs with C, and then G, and then A, and then for some reason there's a G here, and then there's an A, and then again this bond, bond, bond together, and then when the when the enzymes check it, they say, "Oops, there's something wrong here. There's no base pairing here." So the correct way would have been to erase this, right? If you erase that, then you go back to the original setup. But sometimes again, the DNA checking mechanisms fails, and instead of recognizing that that should not be there, instead the DNA would say, okay, there's a G here, it has no pair, so let's insert a C here so that it now has a pair. So now this letter should not have been there. So now that becomes uh, an insertion problem. So let's take a look at it back. So again, look back at it. So you have an insertion where a letter is inserted in the code. You have a substitution where a single instead of A, instead of C, for example, it gets substituted for an A, and you can have deletion where there should be a C there, but it's deleted. So that is from the perspective of individual letters. But you can also have mutation in the chromosome level, because remember, the DNA is a series of letters. I mean, the part of the DNA that codes for the protein is the series of letters. But again, the DNA is so long that it's packaged up in chromosomes. So you could have the next level of mutation, and this is the chromosomal level of mutation. So you could have a deletion where a part of the um, a part of the a part of the chromosome is deleted. So instead of it being there, it's deleted. So a whole section. So if you're talking about a book, for example, page 20 to page 40 is deleted, and you go from page 19 to page 41 immediately uh, without and then the page 20 to page 40 is missing, right? So let's go back at it. So if you look at this part here, um, this part is removed, is deleted. So now the chromosome becomes shorter. Another mutation is duplication, meaning if you have if you have page 20 to page 40, when it gets copied, it becomes page 20 to page 40, and then after that's also page 20 to page 40. So you have a duplicate. And then you could have inversion, meaning if this is page 20 here at the beginning and then page 40 at the last of the segment, it gets inverted where page 40 is in here and page 20 is actually in here. So it gets inverted, right? And then you could have insertion. Now, this is, by the way, for the single chromosome mutation, meaning it only involves one chromosome. A part of it is deleted, a part of it is duplicated, or a part of it is inverted. Now, you could also have mutations which involve a lot of chromosomes. So in this case, for example, it involves chromosome 4 and 20. So what happens here is um, the part of chromosome 4 is actually inserted into chromosome 20, such that now chromosome 20 becomes longer, chromosome 4 becomes shorter. And then you can also have, so technically speaking, from the perspective of chromosome 4, it is deletion. From the perspective of chromosome 20, it is insertion, right? Something is inserted. But in terms of chromosome 4 and 20, that's called insertion because you removed one here and then you transfer it to chromosome 20. Now you can also have translocation. Translocation is where a part of chromosome 4 is removed, a part of chromosome 20 is removed, and then they exchange materials. So this is translocation. Kind of like what happens during crossing over of chromosomes during meiosis, but with crossing over, you exchange the same pages. So page 20 to page 40, page 20 to page 40, you exchange. In this case, it might not be the same pages. So, and again, you are talking about different chromosomes. Remember, in meiosis, you have homologous chromosomes. So it's the same chromosomes, right? And then they're just exchanging the same pages. In this case, you have different chromosomes exchanging different pages. So that is called translocation. Okay? So now we'll proceed with the next part of the discussion, which is, let me remove some bits here first. Um, yes. And then now I'm showing you this is a karyotype. A karyotype is a photograph of all your chromosomes. So in this case, you have chromosome 1, 2, 3, 4, 5 up to chromosome 23. And again, this is 23 because one comes from your father and one comes from your mother. One comes your father. This is chromosome 1 from father, chromosome 1 from mother. They could be exchanged, but you get the idea. It's a pair. And in this case, you have chromosome 23. Take a notice that chromosome number 23, the first pair is longer, so that should be an X. And the next, its pair, its homologous equivalent is shorter, so it's a Y. So this case, we are looking at a male person because it has an XY chromosome, right? So again, just to remind you what is the significance of chromosome number 23. And then we could go to now the disorder. So these are the genetic disorders that people have. So one, you could have what we call cri du chat syndrome. Cri du chat meaning, I don't know how to pronounce that, but I suspect it's cri du chat because it's French. So cri du chat is cry of the cat because people afflicted with this genetic disorder actually have high pitched cries. Okay, so it's like a cat crying. So cry of the cat. And if you notice, the problem here is chromosome number 5, and I hope you can see that one of the homologous pair is shorter. So there's actually some part of chromosome 5 that is deleted. That's why these people have the syndrome Cri-du-Chat, right? And if we were to discuss like what the Cri-du-Chat syndrome, uh, what is its effect? Um, again, they have high speech cry. They have they have individuals with have wide set eyes, small head, jaw, and are moderately to severely mentally retarded and are very short. Okay, so that's Cri-du-Chat. And then after that, we have this disorder. If you notice, chromosome number 21 is weird because chromosome 21 has three copies instead of two. So instead of one from mom, one from dad, for some reason uh, your the chromosome has 23. So this is Down syndrome, right? And if we are to discuss what are the characteristics of people with Down syndrome, this is the most common genetic disorder actually. So you have here flattened nose and face, upward slanting eyes, single palmar crease, short fifth finger that curves inward, widely separated first and second toes, and increase. And basically, the people with Down syndrome kind of they look alike because probably the chromosome that has something to do with the sculpting of the face and the muscle tone underneath the skin and underneath the face is affected. That's why you don't have that individual shape uh, that is akin to the facial muscles of your parents also. So you don't look like your parents. They kind of look uh, similar. People, all people with Down syndrome, right? So that's Down syndrome. And then you have Edward syndrome. And if you, by the way, Down syndrome is called trisomy 21 because it has three copies of chromosome 21. Now this is trisomy 18, Edwards syndrome, right? So if you look at what happens to Edwards syndrome, this is what happens to them. So you have motor and retardation, mental retardation, and about 99% die in infancy. However, those who live past their first birthday usually are quite healthy thereafter, apart from the disfigurement like small mouth, small jaw, short neck, shield chest or short and prominent sternum, and these things. Okay, and then after Edwards syndrome, you could also have something like 11q deletion disorder, which is otherwise named as Jacobson syndrome. So Jacobson syndrome is called 11q deletion disorder because um, the Q part of the chromosome 11 is deleted. That's why it's called 11q deletion disorder. So the Q part is deleted, right? So a portion of the Q part is deleted, and apparently that gives you this characteristics. Either you would have either this person would have normal or mild, normal intelligence or mild mental retardation, but they have very poor language skills or excessive language skills. So kind of the extremes. And they have a bleeding disorder called the Paris Trousseau syndrome, right? So like that. And then after Jacobson syndrome, you can also have Klinefelter syndrome, because and now this is really interesting because instead of, for example, uh, being a woman or a female, which has XX, and being a male, which is XY, these people have XXY. Okay, and so first question: are they males or females? They are males because uh, scientists have decided a long ago how to define male. So male is defined as if there is Y. This is the definition. Female is defined as if there is no Y. Right? So there is no Y, so this is female. There is a Y, so this is male. This one has a Y, so this is male. So again, if we define gender from the chromosome standpoint, if there is a Y chromosome, it is male. If there is a if there is no Y, it is female. Okay, so that's the reason why this is characterized as female. I mean male. So here you notice here, um, Klinefelter syndrome. I will read this one because you might be wondering why this is specified as men with this condition. Because again, the idea is as long as there's a Y, it doesn't matter if it's three chromosomes, XXY. As long as there's a Y, it is considered male, right? So men with this condition are usually sterile, so they can't produce offsprings, and they have longer arms and legs and shy. They are often shy and they have higher incidence of speech delay, right? So as you can imagine, because this XY is the sex chromosome. It has also something to do with sperm production, and so therefore that's why people sperm production because that's the Y. But then it gets hindered because there's another X, right? So therefore that is probably the reason why. That is probably why the reason why they are sterile. So they don't. I mean, they don't. They don't. They're not able to produce offsprings, right? So that's Klinefelter syndrome. And you have Turner syndrome. So Turner syndrome is now again weird because I mean interesting because again XX is female, XY is male. So XXY again is male because again scientists define it as if there's a Y, it's male. Turner's has only X, no Y. But again, because of the definition that we have agreed upon, which is if there's a Y, it's male. If there's no Y, it's female. So this is characterized as female. Okay? And again, that proves my point that the default gender is female because everything has X. It's just that for male, you have a Y chromosome, and at around 6 to 7 weeks, that Y chromosome is expressed, and your female body it's changed to male, right? So, so that's the Turner syndrome. So what happens to people with Turner syndrome? See, Turner syndrome instead of XX or XY, you only have X. I mean, this person only has X. And female sexual characteristics are present. Why? Because again, the default gender is female. So even if you don't have Y, you default back to female. So that's why we characterize this as female. They have a short stature, low hairline, abnormal eye features, and bone development, and caved-in appearance to the chest. So if you ask me what are their machinery, they still have some sort of ovary, because again, the default gender is um, the default gender after all is female, right? So that's it for the genetic disorders, right? So now you might be asking, sir, if there is a trisomy 21, which is Down syndrome, why don't I have why don't we have a disorder called trisomy 3, wherein you have three copies of chromosome 3, trisomy 4, trisomy 5, and so on and so forth? To be honest, I don't know. To be honest, there could be a genetic disorder where it's called trisomy 3, but the reason why you don't have every kind is this: it might be that if you have trisomy 4, for example, it might be that that results to so much alteration that the baby doesn't develop at all, right? So you don't have each kind of this. You don't say if there's 11q deletion, can I have a disorder like 8q deletion where, well, in terms of probability, you could have it could happen, but what is the effect? Maybe the effect is so drastic that it kills the baby, right? So that's why we only discuss these things. This is why we only discuss um, a few, because these are the most common. These are the disorders that we know happen and yet the baby survives. So Cre Down syndrome, Edwards syndrome, Jacobson syndrome, Klinefelter syndrome, and Turner syndrome. Obviously, we could not hope to discuss every single genetic disorder, so we are only limited to discussing this seven or six A B C D E F six disorders, because again, probably these are the most common, and other disorders might be too, again, the effect might be too drastic that the baby doesn't even develop in the first place, right? So again, I'm discussing this because again, I want uh, kind of the logical approach. Like if there's trisomy 21, could there be trisomy 5, trisomy 16? I don't know. There could be, but it could also be that when you have trisomy 6, it could be that the result is so drastic that the baby doesn't develop. That's why we don't get all kinds of trisomy. That's why we don't get all kind of deletion uh, disorders. That's why we only have this six kind of disorders, because that's the idea. That's the concept. Now, I wanted to discuss some things. Where do you get these genetic disorders? Well, we can look at two things: mitosis and meiosis. But with mitosis, remember, mitosis is for body cells. So, for example, these are cells from your skin, from your bones. So, if this, when they divide, for example, the DNA, when it gets copied, it becomes there's a mutation. So, DNA here. So now these are now cells with mutant DNA. It doesn't really make an effect. Why? Because it's just one cell. So, for example, if there are trillions of skin cells and only one cell or only 100 cells mutated, right? After a generation, when they die, that mutation doesn't get passed off, right? Think about it. If I have muscle cells and one of my muscle cells mutated, I mean one of the DNA was mutated. When that muscle cell dies, that mutation doesn't get passed on. It's just like that, right? And since there are many muscle cells, and these mutations are uh, specific to the particular cell in which it occurs, so it doesn't really affect um, the whole family of muscle cells, right? But meiosis is different, because remember, meiosis would affect your sperm and egg cell. So what happens is if you have a mutation and it goes to the sperm cell and egg cell, and unfortunately that sperm and egg cell is the one that that sperm gets to fertilize another egg cell, then the mutations in that sperm cell gets passed on to the baby. Because remember, the zygote is the one is the fertilized egg, and that's the one that divides and become and makes up the body of the baby. So you have a problem there, because if that's defective, then everything becomes defective in that body. But again, that's why mutations in mitosis doesn't matter as much, but in meiosis it matters a lot because it gets passed on to the next generation.